Chapter 22, the Immune System, Part 2, Screencast 3. This screencast will be focusing on the activation and clonal selection of lymphocytes. Clonal selection, this is the process of forming clones in response to an antigen. All formed cells have some TCR or BCR that matches a specific antigen. Now, the antigen challenge, this is the first encounter between antigen and the lymphocyte. This generally occurs within a secondary lymphatic structure. It could be antigen in blood taken to the spleen, antigen penetrating skin transported to a lymph node, or an antigen from the respiratory, GI, urogenital tracts in tonsils, or mucosa-associated lymphatic tissue. Here we're looking at the activation of helper T lymphocytes. And so note, we actually have the MHC class 2 here. And remember, they interact with the helper T cells. And the first thing that's going to happen is the binding with the MHC class 2 molecule. And this will interact with the antigen within the MHC2 class molecule. Now, there also is a second signal, and other receptors interact. Now, this is not shown in this image, and the helper T lymphocyte releases a number of interleukins which bind with the helper T lymphocyte. Now, the result of this is an activated helper T lymphocyte that will proliferate and differentiate to form a clone of activated and memory helper T lymphocytes. So here's where we actually see the concept of memory being introduced. Here's the activation of a cytotoxic T lymphocyte. And notice it's going to be relatively similar to what we just saw, except now we're looking at an MHC class 1 because it's a cytotoxic T cell. The cytotoxic T cell binds with the MHC class 1 molecule of the infected cell, and then the TCR interacts with antigens within the MHC class 1 molecule. As previously, there is a second signal, and this is interleukin 2, which is released from activated helper T lymphocytes, and that activates the cytotoxic T cell. Now, once it's activated, the cytotoxic T cell proliferates and differentiates to form a clone of activated and memory cytotoxic T cells. Here we look at the activation of B lymphocytes. So now the first signal is free antigen binds to the BCR of the B, and the B lymphocyte engulfs and presents antigen to activated helper T cells. Now, the second signal, interleukin 4 released from activated helper T lymphocytes, stimulates the B cells. Activated B cells now proliferate and differentiate to form a clone of plasma cells and memory B lymphocytes. Now, remember, we have the memory cells here, but we also have the plasma cells, and those are the cells that actually will be producing active antibodies. Now here we're looking at the effector response of helper T cells, and what we see here basically is once this T cell has been activated, it's releasing cytokines. In this particular situation, it is interleukin 2. So we can actually see the synthesis and release of various cytokines that regulate the cells of the immune system, and this is the role of the activated helper T cell. Here we're looking at the effector response of a cytotoxic T cell. So now this activated cytotoxic T cell has been activated. It encounters an abnormal cell, and it will result in that cell actually dying. And this is through the process of releasing cytotoxic chemicals that are going to induce apoptosis or death of abnormal cells. Now, most activated B cells become plasma cells. Plasma cells are those cells that actually synthesize and release antibodies. Now, these cells will remain in the lymph nodes. They produce millions of antibodies during a 5-day lifespan. Now, these antibodies circulate through lymph and blood until encountering an antigen. Antibody titer, this is the circulating blood concentration of antibody against a specific antigen. And so it's important that you have enough antibody to respond to the particular antigen that the antibody is specific for. Now here we're looking at antibodies, and these are immunoglobulins. And one thing to keep in mind, I'm not going to spend a lot of time assessing you on antibodies, beyond the fact that they are part of B cells, humoral immunity, and also secreted and synthesized by plasma cells. Now, what antibodies will actually do, they tag pathogens for destruction by immune cells. They are excellent defense against viruses, bacteria, toxins, and yeast spores. They are soluble antigens, so they're combated by humoral immunity. Here we can basically look at an image of an antibody. Typically, they're always going to be described as Y-shaped. We can actually see 2 antigen binding sites and then a constant region as far as the shaft of the Y. Now, the variable regions, that's located at the ends of the antibody arms, and this will actually bind antigens through weak intermolecular forces. The constant region, this contains the FC region, and this determines the biological function. Now, that will be the same in structure for antibodies of a given class. There are actually 5 major classes of immunoglobulins: IgG, IgM, IgA, IgD, and IgE. As noted, you will not be assessed on these, but I'm just kind of talking about them so that you're familiar with their overall function and concept. Now here we're looking at antibody functions, and here we see the binding of antigen binding site of an antibody with an antigen and what it causes. It can cause neutralization. This is when the antibody covers biologically active portions of a microbe or a toxin. We can have agglutination, so the antibody cross-links cells, forming a clump. Here we see that happening with a bacteria. Or precipitation. The antibody cross-links circulating particles, toxins for example, forming an insoluble antigen-antibody complex. And it will just precipitate to the bottom. Now, the exposed portion following antigen binding by antibody will promote complement fixation. So here we see the FC region of the antibody binds complement proteins, and complement is activated. We have opsonization. The FC region of the antibody binds to receptors of phagocytic cells, triggering phagocytosis. Or we actually have the activation of natural killer cells. The FC region of the antibody binds to a natural killer cell, triggering the release of cytotoxic chemicals. Now, here is a list of all of the major classes of immunoglobulins. And I'm going to focus on just 2, IgG and IgM, because we're going to see them in a little bit when we talk about memory. And so where we're going to find IgG is in body fluids, including blood, lymph, cerebrospinal fluid, serous fluid, peritoneal fluid, and breast milk. Their actions is basically neutralization of viruses, bacteria, and toxins. They work via agglutination, precipitation, and the activation of complement. They also will opsonize natural killer cells and activate, and so they're antibody-dependent cell-mediated cytotoxicity. They have a relatively long lifespan of about 23 days, and these are actually used for passive immunity and crosses the placenta. Whereas IgM, we're looking at a B lymphocyte receptor in breast milk. Neutralization, agglutination, complement binding are jobs associated with this. They only last for about 5 days. Now, this is the first produced antibody, and it is the only antibody produced in a fetus.